Okay, we're going to start with the second part of Module 1 lecture. I hope you can see this well. Okay, so we're going to talk about homeostasis, okay? And what is homeostasis? This is a state of dynamic equilibrium in the body with its respect to its internal environment and functions. So basically, just to simplify this whole thing, it's equilibrium, this is the definition, equilibrium in the body with respect to the environment. Now, if you just know that this is equilibrium in the body, that should be more than enough. Okay. So what is equilibrium? Equilibrium means balance or stability, okay? So what it's doing is basically your body is trying to maintain homeostasis or equilibrium no matter what's going on around it, okay? It tries to stay steady so that you can breathe, so that your blood can flow, so that your blood pressure is good, all of those things. Okay, now we're gonna learn that stress, okay, stress can cause the body to move away from equilibrium or homeostasis, okay? Now, what's an example of this? It's fever when you're sick. Fever tries to maintain your internal body temperature. When your body temperature goes up due to illness, you get a fever to help bring that temperature down, okay? So we're going to talk about how this homeostasis is maintained. So the control of homeostasis is due to feedback systems. I'm not going to go too much into this. It's going to be a very bare explanation of it. You'll have to go into the textbook to really read into this, which starts on page 9 in the textbook and goes on to page 13 or so. Okay, now there is something called the control center. Now this control center it's a part of the body. It can be either the CNS, which is central nervous system, okay, or the endocrine gland that receives some information, determines what that is, and signals a response to correct anything that causes the imbalance in the body. So this receives info and signals a response. Okay, now let's talk about an example of this would be blood pressure, okay? So blood pressure shouldn't be too high or too low. A good blood pressure causes the blood to flow through your heart and it also helps control the circulation. Now, our blood pressure in the body, okay, I'll put an example as blood pressure and I'll abbreviate that as BP. Blood pressure is detected by receptors, which is located in the arteries near the heart and in the neck. Now, what happens is when the receptors in these areas notice that the blood pressure is going high, these nerves that are associated with that area send a message to the brain, which we said, the CNS contains the brain, the control center, indicating that your blood pressure is too high. Now, when this CNS brain or endocrine gland receives this information, what does it do? Okay. In this case, in blood pressure, the endocrine gland is not involved, but, well, not for basic knowledge purposes. But what happens is there's a receptor that monitors things in these areas, okay? So that receptor receives this info, signals a response through the brain to correct this blood pressure. Okay, so let's talk about a receptor. It's a structure in the body that monitors the body's changes or variables. Now, the signal's the brain, but the brain can't directly lower your blood pressure just by itself, okay? So what it does is it sends, the receptors in the brain gets a message that, hey, blood pressure is too high. So what does the brain do? It sends a message. That's what the brain does. It sends a message via nerves, which targets an effector. Now, what is an effector? It's a structure in the body that can change the value or can change the variable in response to a signal. So basically, it can change the problem in response to the signal from the control center. Okay. So this effector organ, or this effector, can change the blood pressure. So what happens is the brain sends a message via the nerves to the effector, and the effector then lowers the blood pressure. The effect is called the response. What's the response? Response is that the blood pressure normalizes. Okay. Now, what is this? This whole thing is known as the negative feedback system. Okay. Now, if you look on page 12, it'll explain to you how that works. Figure 1.6. So pretty much what this whole negative feedback system does is it maintains homeostasis, okay? Now, there's other examples here given in this chapter in the book. You can just do a quick skim through it just so you have a more thorough knowledge of this. It would be helpful, okay? Another example I'm just going to say very briefly is the hormone insulin, and that's where the endocrine gland is involved, is an example of that. Insulin lowers the blood glucose. So when your body has high blood sugar levels or glucose, the receptors are signal that, hey, there's too much blood glucose in the body. So what does it do? The body triggers the endocrine gland to send a response saying, hey, we need insulin. Our body needs insulin. So then insulin is released to lower the blood sugar. So that's an example of negative feedback system. Okay, next we're going to get into the cell structures, okay? Some of this might be review for you guys because you must have done this in life science or in past classes, probably pre-biology, or even if you've taken general biology, it'll be in there. You would have covered some of the stuff there as well, okay? So we're going to talk about cell structure. and organelle function. Now, I've already explained in the last lecture that our body has organs. It consists of organs that work together to put all the functions together that causes a human body to function. Now, inside of a cell, each individual cell has its own so-called quote-unquote organs as well, and they're called organelles, okay? And that's what we're going to be talking about. Now, what we talk about today in this particular, this is the basis of all of biology, and you'll have to know how to draw this picture, okay? I'm going to put it up so that I'm not wasting time drawing and wasting your time, so I'm just going to put up the picture here. I think you can see it. Okay, and we're going to talk about this one by one. Okay, now if you look on page, if you look on page 14, figure 1.7, you'll see the structure. I mean, it's much more fancy in the book, but I made it in a way that you can easily draw it. So let's start out with talk about the beginning, from the beginning, what we include in the cell. Okay. Now, first of all, the cell has something called a plasma membrane. Okay, right here, that's the plasma membrane. Okay, this whole circular wall. What does the plasma membrane do? It pretty much holds in everything in the cell. Okay, our organs are made up of tissue, and tissues are made up of cells. So there's many cells that contain these organelles in our body. Okay, now this plasma membrane is the boundary of the cell. Okay, holds it all together. It has many receptors on these cells. Okay, we talked about receptors. And these receptors not only carry signals, it also determines what substances are going into the cell and what can be allowed to go out of the cell, okay? And it has to go through the plasma membrane to get in or out of the cell. Okay, so the next thing you need to know is the nucleus, okay? Now, this circular structure is the nucleus. And in the center of that is the nucleolus. Okay? Now, the nucleus is wrapped in a nuclear membrane called the nuclear envelope. See this? This is a double membrane. So you see two layers here. That's a double membrane known as a nuclear envelope. Okay, so that's what this is. And inside that double membrane, that's the whole entire thing is a nucleus, and in the center of that is a nucleolus. Now, this nucleus contains genetic material, okay? And this is DNA. You guys know it as DNA. I know you've heard of that before. The DNA is known as deoxyribonucleic acid, okay? Now, this within the and also within this, you get RNA, okay? So, now we'll talk more about cellular reproduction as we go on in this chapter. But what the DNA does is, during cellular reproduction, the DNA forms chromosomes, okay? That's the purpose of the center of this. Now, we're going to go over that soon. Now, the nucleus can be thought of as a control center of the cell, so pretty much the brain of the cell. Why? Because it contains DNA. DNA is there, and that's what determines a lot of the functions that go on here. Now, the DNA codes for all the proteins that the cell produces. Okay, so without DNA, there's not going to be any proteins. Now, between the nucleus and the cell membrane, okay, so we have the cell membrane. The cell membrane is also known as a plasma membrane. It's pretty much the same thing. Now, in between this nucleus and the cell membrane, or the plasma membrane, you have something called cytoplasm. So all of this, everything that you see here, is cytoplasm, okay? Now, the fluid part of the cytoplasm is known as cytosol. So basically this whole cell is kind of almost like a watery gel-like consistency, and all these things that you see in it are floating around in that, okay? And the cytosol, which is the fluid part, contains many chemicals. It includes ions, proteins, molecules, okay? And what it does is there's a lot of processes that goes on in the cytosol, such as breaking down of sugar and fats and formation of other chemicals and other things the cell will need. Now, if you think of the nucleus as the control center of the cell, the ribosomes, okay, let's get it. Let me see if I've drawn that here. These ribosomes, okay, are, what it does is it is kind of thought of as the working point of the cell, or the tiny kitchens of the cell. Proteins are synthesized in the ribosomes, okay? I would also like you to refer, so there's these little dot-like structures around. See these free polyribosomes? These are in blue here. I hope you can see them on there. These are ribosomes. So the ribosomes are an essential part of the cell, okay? So they're really, really tiny, and that's why they're like little dots. Now, sometimes the ribosomes are alone. And when you see them alone, see all by itself here, these are known as free ribosomes, okay? That means they're free, they're not attached to anything. Now, we're gonna talk about the endoplasmic reticulum, okay? Now, if you look here, where do we have this? Oh, yeah, labeled it here. I'm gonna make it darker so you can see it. This is the endoplasmic reticulum. Okay, and hopefully this brightens it up. Okay, that's how you draw the endoplasmic reticulum. Now, if you look at this, there's a smooth endoplasmic reticulum and there's a rough ones, okay? The smooth ones, the smooth part of it does not have any ribosomes on it. What makes it rough is the ribosomes. So all these blue dots you see here, that makes it rough endoplasmic reticulum. And when you don't have any of that on there, it's smooth, okay? Now, the smooth endoplasmic reticulum is basically, it allows for intracellular transport, okay, within the cell. It also produces lipids and carbohydrates. Okay, so smooth, you can think of smooth fat cells, which are known as lipids. So any fat cell is known as a lipid, and carbohydrates. Now, rough endoplasmic reticulum, on the other hand, is also used for intracellular transport, but it's rough in appearance. Why? It looks rough, right? Because these bumpy little ribosomes are all over it. Now, since we said that ribosomes are used to make proteins, that is what the rough endoplasmic reticulum does. It is used for protein synthesis and transport of these proteins, okay? So the smooth, what does it do? Lipids and carbs synthesize, and it also breaks down toxins, and rough one is for protein synthesis due to the ribosomes, okay? Now, if you see how much smooth or how much rough endoplasmic reticulum there is, that's what let's say there's more smooth endoplasmic reticulum than rough in a cell. That means the main function of that cell is more so to make lipids and carbs. If it has more rough endoplasmic reticulum, it's more, the major function of that is more to make proteins, okay? Now, so here we have a couple of other things here. So now, let's go on to the Golgi apparatus and we'll come back to this to explain this a little bit more. Before I move on, the endoplasmic reticulum also is known as the assembly plant, okay? Because that's where everything's assembled. If you look at it, the way it's pictured, just imagine an assembly plant in a factory, pretty much they sit and they make one part, pass it on. The next part makes another part, keeps passing it on back and forth until it's formed. Okay. Now, the Golgi apparatus. Let's talk about that. Do I have that in here? Okay, so the Golgi apparatus, let me get a marker, is more wider and more ribbon-like, okay? So it's more like this. Kind of similar structure, but it's wider. This is more narrow, and this is the Golgi apparatus. So this is known as a cell's packaging plant. Okay. So what it does is it takes all these chemicals, packages them, mainly for secretions, okay? They do a lot of chemical modifications within this Golgi apparatus, okay? The best way to think of this is as a wide ribbon. It's a ribbon-like structure, okay, because it's that little thicker. Now, nervous system cells, they're known as neurons. They have a lot of this Golgi apparatus because they do a lot of cell packaging, okay? And it secretes chemicals. For example, when you eat, right? Our salivary glands secrete saliva. What makes the saliva? The Golgi apparatus. There's Golgi apparatus within the salivary glands, and that's what produces saliva. Now, the secretory vesicle that we see here, so this is seen at the end of the Golgi apparatus. See the secretory, this little black tiny circle? That's a secretory vesicle, okay? It comes from the Golgi apparatus. Now, what it does is once this Golgi apparatus packages the chemical for secretion, it puts a chemical into this little secretory vesicle, okay? This vesicle then pinches off from the end of the Golgi apparatus, okay? And what it does is it travels through the cytoplasm, okay, to the plasma membrane. So here we are at the edge of the plasma membrane, where it all of a sudden it releases the contents out of the cell. Okay? But it doesn't just randomly do this. It needs to get a signal from the body showing that this particular chemical needs to be released. So again, we come back to that feedback system, secretory vesicles are not just randomly secreting secretions. It depends on what the signal is. For example, your mouth is just not constantly going to be drooling or constantly your mouth is always moist with saliva, yes, but when do you see excess saliva? It could be when you, at the sight of food or the smell of food, it triggers the signal like, oh, your body may be getting ready to eat, so it secretes more saliva. So that's an example of that is when the secretory vesicle will secrete the fluid in response to that signal. Okay? So next, let's talk about lysosomes. Okay, and that's here. So a lysosome, this is pretty much the structure of a lysosome. It's a little circle with two little thingies at the end of it. Now, the lysosome is also, it's a kind of vesicle, okay? Its main function, what does it do? It breaks down lipids, proteins, and polysaccharides, and carbohydrates, and nucleic acids. What are nucleic acids? Nucleic acids are RNA and DNA. So the lysosome breaks it all down, okay? Now, for a lysosome to do its job, it needs something called enzymes, okay? So what does enzyme do? An enzyme breaks down a substance to make it less dangerous. Sometimes it breaks down substances and makes it more dangerous, okay? So what you need to know about lysozyme is pretty much breaks down I'm going to put lipids, L, proteins, carbohydrates, and other stuff. Okay, it gets, basically the importance of this is it gets rid of damaged tissue. And that's the main thing you need to know about it. That's why it's breaking all this down. Because if damaged tissue accumulates, we start developing diseases. Okay? Now, I know you've all heard that, you know, if our brain does not, if we don't get, or our heart, any part of our body does not get oxygen for a certain amount of time, we die. We need oxygen to live and survive. So without oxygen, our tissue or our cells can live maximum 4 to 8 minutes without oxygen, depending on which tissue type we're talking about. But in general, 4 to 8 minutes. So what happens is after 4 to 8 minutes without oxygen, the lysosomes of the neurons can't hold themselves together, okay? They land up bursting. So these lysosomes, in order to function and maintain their structure, it needs a constant supply of oxygen. And we have lysosomes all over our body in every single cell. So once the oxygen stops, it bursts, okay? And then it since it's breaking down all these toxins, all those toxins are released. And this kills the entire neuron or the nerve cell. And this is what leads to pretty much brain death, okay? It kills the neuron. So what does that mean? That means if the oxygen supply is cut or diminished to any tissue or organ, the goal of the hospital or the doctor, or the goal is to make sure that that tissue somehow is getting oxygen before it completely dies, okay? So now sometimes, okay, ruptures of this lysosome is a good thing, okay? So if there's like damaged lysosomes, for example, they will self-digest. Why? Because it takes away, again, it's the system in our bodies that are taking away all the dead or bad cells in our body, okay? Now, let's talk about another example of this. Think of a cut, okay, an infected cut. Let's say someone gets a bad cut while they're doing work on their lawn. Let's say that cut is not kept clean and it keeps getting dirty and bacteria multiply. What happens? It produces something called pus. And I'm sure all of you have seen pus before. That pus is from white blood cells, okay, which we'll talk about later on. Now, what happens is these white blood cells have burst their lysosomes, okay? And so why does it do that? Because it bursts because it kills the white blood cell, which is a cell that's trying to fight the infection, but it also kills the foreign invader or the bacteria, okay? And so the lysosome breaks open to kill that bacterial cell. So it can do it for good, and it also can cause bad things if it's due to lack of oxygen. Okay. Now, we're going to talk about mitochondria. Okay. Now, this is very important because mitochondria is something we are going to talk about later on too. Okay. So it's an oval structure with this, again, a ribbon-like structure inside. Mitochondria is where ATP is formed, okay? ATP is adenosine triphosphate, okay? This is the powerhouse of the cell, okay? All energy comes to the cell and to the body in the form of ATP, and this is produced in the mitochondria. Now, you'll have to know this whole cell, you'll have to know, be able to draw it, label the structures. So this is something you should practice doing before the test, okay? Now, the cellular energy is stored as ATP, okay? That the majority of the ATP is made here. There are other areas where it's made too, but for right now, you need to know that this is where ATP is formed, okay? Now, Now, the other thing is, so mitochondria also contains mitochondrial DNA. Okay, DNA mainly is the nucleus. We talked about that, but it's also, there's something called mitochondrial DNA. Okay, so to a certain amount, certain degree, it can produce its own proteins, but that's not its major function. Now, one thing about mitochondria, why do you know about ATP? Because anytime you have reduced energy or reduced ATP, usually it's when your body starts feeling tired and stressed or sick. So you can attribute this to pretty much any illness. So let's say if somebody has the flu, okay? Now what's happening? All the energy in the body, the majority of the energy in the body, the ATP, is used for energy for all the cells to function and fight that infection. Now what happens? So that depletes ATP in your body, causing you to feel tired and sick and not feeling energetic at all. So this is why people don't feel well when they have a cold or a sinus infection. It's because of decreased amounts of ATP giving energy that's needed. Okay? Now, what else do we not cover here? Okay. The next, I don't know what we talked about. So, we didn't talk about centrioles. So centrioles are these little structures that look like, let's see, like kind of like these, okay? Like a tube, pretty much. So centrioles are found in the centrosome, okay, which is the center of the microtubule formation for the cell. Now, microtubules, you can see this on page 16 in your book. They're like rope-like structures, and they're strands of proteins that form the structure. Now, what does centrioles do? They influence the movement and shape of the cells, and they're important in cellular reproduction, okay? Now, we should also talk about cilia. Cilia are these little hair-like projections outside of the cell. Not all cells have cilia, but some of them do. And they're like tiny little hairs, okay? And what is it? It's an intricate arrangement of microtubules. They use these centrioles, okay, to make that cilia, okay? And for example, cilia prevents infection from coming in. It helps things to move along. So for example, it's in the back of our nose, in our trachea, in our airways. Okay, what does it do? It pushes, for example, it beats upward and it pushes mucus towards our throat, like when we're sick or when we need to spit something up. It kind of traps the bacteria or dust particles and gets it out of our body and kind of cleans it up. Okay. Now, without the cilia, this mucus cannot move back up into our mouth or wherever, or our nose, so that we can blow it out or spit it up. Okay. Okay. So what you have to know, just so here's a breakdown of it. So the lysosomes, and we just went over this, you can just take this down in your notes. So the lysosomes release enzymes to break down lipids, proteins, polysaccharides, carbs. It needs oxygen. If they don't, it bursts, kills a neuron. If it's ruptured, it also gets rid of diseased or damaged tissues. The next thing is the centrioles, is what this orange structure here shows. And they're found in centrosomes. And the center of microtubule formation. That's what the centrioles do. It forms spindles. It influences movement in the mitosis. We're going to talk about mitosis and meiosis and the shape of the cell and also cellular reproduction. Now, why do you need to know the microtubule formation? Because it forms cilia, okay? And then the microfilaments cause movements, enable cells to contract. The muscle cells contract and relax, and the microfilaments are what allows the cell to do that, okay? Okay, again, I'm going to put this down so you can just copy the notes. Mitochondria, you'll need to know this is the major site of ATP synthesis. It is the powerhouse of the cell. This is how it's drawn, okay? Some ATP is made in the cytoplasm, okay? First stage of cellular respiration known as glycolysis, which you'll know for later. Just keep it in your notes because you might need to refer back to this when we get on to those chapters. Most DNA is stored in the nucleus, but some DNA is in the mitochondria as well. And the mitochondrial DNA is known as mitochondrial DNA. It codes for certain proteins to make the mitochondria function. Now, the DNA plus the ribosomes present in the mitochondria, this enables mitochondria to produce some proteins, but not everything. It can't produce all the protein it needs. So some proteins are vital to mitochondria. It's still produced by the nucleic DNA, which is, obviously in the nucleus, and the ribosomes in the cytoplasm, okay? The proteins then are transported to the mitochondria, the ones that are needed, okay? Now, okay, so we're going to talk about, so that was the structure. I'm going to move on to proteins. Okay, these are large molecules formed by joining of amino acids. And I'm going to put amino acids, from here on I'm going to abbreviate it as AA. Okay. Now, let's talk about enzymes. These are catalysts. They're molecules that speed up a chemical reaction. That's what a catalyst is, and that's the activity that enzymes do. They speed up the chemical reaction, such as breaking down or changing one substance into another. Okay, another protein is hormones, and that has its own chapter, so I'm not going to go into that. Just know that hormone is a protein. And then antibodies are proteins, okay? And you'll need to know that as well, and that will go on later on too. We don't really get into that right now. Okay, now we're going to talk about protein synthesis. Okay. Okay, now there's 2 steps to protein synthesis, okay? One is transcription, and the other is translation. Now, what is protein synthesis? What's the goal of this out of these 2 steps? The goal is basically gene expression. That's what the goal of protein synthesis is. Okay. Now, two things. Well, this is a couple of things actually, but I guess I should start at a different page for this, but I'm going to give you some generalized things about this, and then we'll go on to an actual chart of transcription and translation as we get more into this, okay? Now, let's talk about transcription first. Now, the goal of transcription is to make RNA copies. Some of this is going to be memorization after you understand it, because you will need to know the difference between transcription and translation, and you'll need to know these are the two steps of protein synthesis. So this is actually important, okay? It takes info. Transcription, what it does is it takes info, takes info from genes in our DNA to make proteins. Okay, now, how does it do this? It makes a copy of a gene's DNA sequence to make an RNA molecule. So it kind of, it takes it and kind of photocopies it. Of a gene. So how does it do this? It does it by enzymes. Now here, what are the enzymes here? RNA polymerase. Now, anytime you see an ase at the end of a word, that means it is an enzyme. That's how you identify enzymes for the most part. Here and there are a couple of enzymes that don't end in ase, but overall, if it ends in ase, it is definitely an enzyme, okay? Now, this RNA polymerase, what does it do? It links nucleotides to form an RNA strand. So basically, what is it doing? It's using a DNA strand to make a template. Okay. Now there's 3 steps of this. You don't have to know each one what it does, but you got to be able to label the 3. Okay. And 1 is initiation. 2 is elongation. 3 is termination. Okay. Now the products it makes, what is the products of transcription? It's mRNA, tRNA, and rRNA. So what is this? mRNA is messenger RNA, tRNA is transport RNA, and rRNA is ribosomal RNA. You'll need to know that. mTRNA and what it stands for. Okay, so now we're gonna have to go onto a different paper just to get all this, and we're gonna go over this again too. So I will just leave that there. Again, I'm gonna split the paper into transcription. If you guys have enough space on your notes, just you can just keep it going, but. Okay, so we're going to talk about the site of where this stuff occurs. Now, the site in transcription is the nucleus, and in translation, the site is cytoplasm. Okay. Now, so if we just talk about RNA by itself, the way to make it functional is to add that, make it a messenger, transfer, or RNA, okay? That's the end product, okay? That happens in the nucleus and known as transcription, okay? Now, let's talk about the purpose here. So the purpose here is to make functional RNA. And by functional, again, I'm going to say it, mRNA, messenger RNA, transport RNA, and ribosomal RNA. These are the end products of transcription. There's a lot more information about transcription than translation because it's a longer process to make all this stuff, okay? Now, again, we're going to talk about initiation. So what happens here? I mean, if you read in the book, it's a lot more information, but I'm just giving you a simple synopsis. Okay, initiation, what happens? The RNA polymerase, again, polymerase is an enzyme. So the RNA polymerase binds to the promoter in DNA. What is a promoter? It directs the exact location for initiation of transcription. The second step is elongation. Okay, here it's unwound and produces a complementary RNA strand. But in the opposite direction. So what happens to the DNA after this? So this termination pretty much is the DNA rewinds to a double helix. What's a double helix? It looks like this. Or something like that. It's not a good picture, but you can see it in your book. Okay, that's how it ends. Okay, and then the products of this is mRNA, tRNA, rRNA. Okay? Now, So if you look at the picture, let's go back to this. So basically, your messenger RNA, the mRNA, carries this to the rRNA, right? To make the proteins, and then that's going into transport RNA, which carries it in here. So you're taking that messenger RNA to basically to the nucleolus. Okay, now let's talk about translation. So basically, what is this? It's a protein synthesis from an mRNA template. What is that? Messenger RNA. So how does this work? The mRNA is transported to the rRNA, where it's in the assembly plant to produce a protein. And what is the assembly plant again? The endoplasmic reticulum. Okay. Now, so that's transcription and translation, okay? So, one thing you need to know, you need to know DNA has its own alphabet, okay? Except that DNA has 4 letters. Okay, so let's start another page. Okay, so four letters, otherwise known as bases. Okay. Now there is adenine, abbreviated as A, thymine, abbreviated as T, cytosine, abbreviated as C, and guanine, abbreviated as G. Okay. Now, if you take groups of 3s of this, any 3 of these makes a gene. Okay. But basically they have 4 letters, 4 of these substances, 4 of these proteins to work with. Okay. Now, let's talk about the RNA. Now, in RNA, you have uracil. You need to know that. So, you need to know this. Okay, so so the next thing that we're going to learn about is mitosis and meiosis. That's a lecture on itself, so that will go on for next week. Okay, that's it for this lecture.